The cell is the smallest unit of life. The cell was culled from Latin word called cella, which means small room. It represents the organic, functional and structural unity of every living organism, and it forms the basis on which the fundamentals of biology is built. They are also referred to as the building blocks of life. Cells are made up of a cytoplasm covered by a cell membrane, and the cell membrane is made up of many biomolecules like nucleic acids and proteins. Some organisms are unicellular, while some other ones are multicellular. The unicellular has just one cell, while the multicellular organisms have multiple cells. Good examples of unicellular organisms are bacteria, while plants and animals represent multicellular organisms. There are numerous plants and animals around, and each of them varies in the number of cells they have as constituents. In human beings, for example, there are up to 10 trillion cells. Most of the cells in plants and animals can be viewed easily using light microscope, since their size is between 1 and 100 micrometers. Cellular levels. The smallest unit in cellular level is the atom, which is composed of elements like nitrogen, carbon and hydrogen. This is followed by molecule, which is composed of molecular structures like amino acids, glucose, fatty acid and the likes. Molecules are a combination of atoms. Molecules, on the other hand, combine together to form macromolecules, and good examples of this are the DNA. Macromolecules combine to form organelles, a good example of which is the nucleus. Others are mitochondria, ribosome, cell membranes, lysosome and so on, with each of these organelles playing specific roles in the structure and functions of the living cell and the organism. Group of organelles combines to form the cell, and there are different forms of cells. Good examples are the nerve cells, which is located in the brain and spinal cord and only found in animals. Conclusively, atoms, molecules, macromolecules, organelles and cells combine to make up the cellular level of life. Organism level. Cells combine to form what is called tissue, and a good example of tissue is the nerve tissue. Others are connective tissues, epithelial tissue and muscular tissue. These 4 types of tissues form the main types available in living things, especially animals. Tissues combine to form organs. There are various types of organs in the body, and good examples of organs are brain, eyes, liver, heart, kidney, and many others. Organs work together to form systems. Magnificent examples of organ systems are nervous system, which involves various organs like the brain. The eyes are classified as an extension of the human brain. Other systems are the respiratory system, involving the nose, the lungs, and even the heart. There is also the reproductive system, the digestive system, with each playing its specific role to ensure physiological balance in animals. The organ systems combine to form the organism like birds, man, lion, and so on. Types of cells. Cells can be classified broadly as prokaryotic cells and eukaryotic cells. Prokaryotes. They are minuscule cells that are surrounded by the plasma membrane. They also possess characteristic cell wall, and the cell walls may differ in their compositions, which are determined by the particular type of organism they represent. Prokaryotes do not have a nucleus. They only possess linear or circular DNA structure. Besides, they lack many other membrane-bound organelles, including mitochondria and lysosome. However, they possess ribosomes. Their protoplasm is made up of the cytoplasm and the chromosomal region. They possess flagella, which propel from the external surface. The number of flagella differs from one cell type to another, and the flagella function in motility. Additionally, they possess 6 pili on their external surface, which are used for attachment. The chromosomal region appears just as fibrous deposits when viewed under the microscope. The two domains of prokaryotes are bacteria and archaea. The general characteristics of the prokaryotes are highlighted below. First, they are small. Second, they are simple. Third, most of them do not possess membrane-enclosed organelles. Fourth, good examples are bacteria and archaea. Fifth, their DNA is circular. Sixth, their type of nucleus is called nuclei region, not true nucleus. Seventh, the ribosome is 50S and 30S. Eighth, their cytoplasm have very few structures inside. Ninth, they use flagella for movement. The flagella is composed of flagellum. 10th, they have no mitochondrion. 11th, no chloroplast. 12th, cell division is by binary fission, which is very simple division. 13th, they have only single chromosome. Eukaryotes. Just like prokaryotes, eukaryotes too are surrounded by the plasma membrane. They possess a nucleus, on the other hand, which is made up of nuclei surrounded by nuclear envelope of membrane. Also, eukaryotes have many membrane-bound organelles like vacuoles, rough and smooth endoplasmic reticulum, lysosome, chloroplasts, and mitochondria. They contain organized chromosomes too, in which are stored genetic materials. These cells differ in shape, structure, appearance, and function. In multicellular organisms, one can find up to 150 different types of cells, with each having its individual function. Other living things possessing cells are protoctists, fungi, and plants. They only possess a maximum of 20 different cell types. Many of these cells have cilia, called primary cilia. This type of cilia is important in thermosensation, which enables the cell to sense change in temperature, mechanosensation, which enables the cell to sense contact, and chemosensation, via which the cell senses change in chemical concentration. These are the features that make primary cilia to act as sensory cellular antennae. It can coordinate several cellular signaling pathways, and it can couple ciliary motility to signaling pathway. Some eukaryotes that are motile can move around using either flagella or cilia. Some plants do not contain any form of motile cells. Examples of such plants are flowering plants and conifers. Many complexities are, however, noticed in prokaryotic flagella than in eukaryotic flagella. The general characteristics of the eukaryotes are highlighted below. First, they are larger. Second, they are more complex. Third, they contain membrane-enclosed organelles. Fourth, examples are animals, fungi, plants, and protists. Fifth, they have a true nucleus. The DNA has a linear molecule. It is made up of chromosomes with histone proteins. Sixth, RNA synthesis takes place in the nucleus while protein synthesis occurs in the cytoplasm. Seventh, the ribosome is 60S and 40S. Eighth, the cytoplasmic structure is highly structured by cytoskeleton and endomembrane. Ninth, it makes use of flagella and microtubules for movement. The microtubules contain cilia. Some of them make use of filopodia and lamellipodia for movement, both of which contain actin. 10th, one cell can contain several thousands of mitochondria. 11th, algae and plants contain chloroplasts. 12th, the organization varies from single cells to colonies and higher multicellular organisms that have a specialized cell. 13th, cell division is by mitosis and meiosis. Somatic cell division is by mitosis, while sex cell division is by meiosis. Mitosis can be either via budding or fission. Dobre, 13. Cell division is by mitosis and meiosis. Somatic cell division is by mitosis, while sex cell division is by meiosis. Mitosis can be either via budding or fission. 14th, it contains various chromosomes. Cellular growth and metabolism. Cellular metabolism can be defined as the process in which cells process the nutrients it receives. Metabolism can be classified into anabolism and catabolism. Anabolism is the process whereby cells carry out various biological functions and produce complex molecules using reducing power and energy. Catabolism, on the other hand, is the process in which cells produce energy by breaking down complex molecules. When the organism consumes complex sugar, catabolism kicks in to break the complex sugar into a simple sugar that can be absorbed and utilized by the body in biological processes. These simple sugars are called monosaccharide, and a good example is a glucose. Fructose is another type. While glucose consists of 6 carbons, fructose consists of 5 carbons. Once glucose has gained entrance into the cell, it is broken down and used in producing adenosine triphosphate ATP. Production of ATP occurs via 2 different pathways, which are: first, replication, second, protein synthesis. Replication occurs in living things via division of a single cell to 2 daughter cells. The primary single cell is called the mother cell. This is the process via which cells multiply. Cell division results in vegetative reproduction and tissue growth. Vegetative reproduction occurs in unicellular organisms. Cell division in a prokaryotic cell is called binary fission. It has some similarities to cell division in eukaryotic cells, which can either be mitosis or meiosis. In mitosis, the cells divide into 2 equal daughter cells, which have similarity to the mother cell. In meiosis, on the other hand, a diploid cell, acting as the mother cell, divide into 2 cells, either of which has half of the number of chromosomes in the mother cell. The daughter cells are called haploid cells, half of diploid. Formation of haploid cells is not the end of meiotic cell division. The haploid cells now combine with other haploid cells to form gamete. Meiotic cell division is undoubtedly a more complex process than the mitotic cell division, which is somewhat straightforward. DNA replication occurs during meiosis I, but no DNA replication in meiosis II when cell division occurs the second time. Protein synthesis. Proteins produced by cells are used to maintain and modulate cellular activities. Protein synthesis begins from an amino acid, which is the building block for protein. The 2 major steps involved in protein synthesis are translation and transcription. In transcription, complementary RNA strand is produced using genetic information in DNA. The RNA strand produced then undergoes processing to produce messenger RNA, mRNA. Messenger RNA then binds to the ribosome, protein RNA, which is located in the cytosol. The mRNA is then translated to polypeptide sequences. Chromosomes. Chromosomes are responsible for carrying genetic information. They are composed of long strands of DNA referred to as genes. Human beings have up to 23 pairs of chromosomes. 22 pairs out of the lot represent autosomal chromosomes, while the last pair represents sex chromosomes. The single pair of a sex chromosome in the female is XX, while the single pair in the male is XY. At conception, the father donates 1 haploid chromosome, while the mother donates the second haploid chromosome. DNA. It is an acronym for deoxyribonucleic acid. It is made of genetic information that leads to the formation of an organism. They have a spiral shape, just like a spiral staircase, and they are called double helix. The DNA is made up of DNA bases, which are 4 in number. They are adenine, A, thymine, T, guanine, G, and cytosine, C. These genes are combined in a specific pattern, and the pattern of combination indicates the kind of protein that will be produced. If any of the patterns is substituted with another DNA base, the type of protein produced will also change. This is the basis on which genetic modification and molecular biology are built. The type of protein produced by the cell will determine the function of that cell. Cellular organelles and functions. Cellular organelles are divided into two main types, which are membrane-bound organelles and non-membrane-bound organelles. Examples of membrane-bound organelles are vacuoles, secretory vessels, peroxisomes, lysosomes, Golgi apparatus, chloroplasts, mitochondria, endoplasmic reticulum, and nucleus. Examples of non-membrane-bound organelles are ribosome, microfilaments, microtubules, intermediate filaments, cell junctions, centrosomes, flagella, cilia, and pili. Cell wall. It is located on the outer layer of the prokaryotic cells and plant cells. It is composed of chitin and cellulose. It is, however, absent in animal cells and some prokaryotic cells. It functions in support and protection from the external cellular environment. Cytoskeleton. It is a network of protection filaments. They function in chromosome movement during the process of meiosis and mitosis. They are involved in the cytokinesis process of cell division. They function in protozoa to discharge contents of vessels for water regulation. They are equally involved in pigment vessel movement for protective coloration. They are involved in cytoplasmic streaming and are equally featured in movement of vessels between the cell surface and organelles. Nucleus. It is the center of genetic information and it plays an important role during cell division. It is involved in the transfer of character from one cell to another cell. Endoplasmic reticulum. It can be rough or smooth endoplasmic reticulum. The smooth type has no ribosome on it, while the rough type got its name from deposits of ribosome on its outer surface. The rough type is involved in protein synthesis, and both types are involved in intracellular transportation. The smooth type is involved in protein transport and protein folding. It is involved in other processes like glycosylation and formation of a disulfide bond. It helps with the modification of drugs via cytochrome P450 enzymes, especially in liver cells. Mitochondria. It is referred to as the powerhouse of the cell. This is because it generates energy in the form of ATP, which is the fuel required for cellular activities. It is involved in apoptosis and regulation of cellular metabolism. It equally synthesizes some steroids. Chloroplast. It is only available in plants and it is responsible for the production of chlorophyll, which is responsible for the green coloration of plants. It is the site of photosynthesis, a process by which plants convert carbon dioxide and water to glucose and oxygen. Golgi apparatus. It is involved in intracellular transportation. It is involved in the modification, sorting and packaging of macromolecules for secretion of delivery to other organelles. Lysosome. It acts as a store for various enzymes like proteolytic enzyme, which can digest intracellular and extracellular substances. They enable cells to utilize nutrients. They are involved in biosynthesis, which is the recycling of unwanted products to other materials, heterophagy, which involves digestion of extracellular materials, and autophagy, which involves digestion of intracellular materials. Cell theory. Cell theory is a scientific theory that gives an in-depth description to the properties of cells. The cell is the basic or fundamental unit of life. It is equally the fundamental unit of reproduction. Many cells are not visible using naked human eyes, but they can be clearly seen using microscopes, have a history behind them, but they have been improved over time since the first type of microscope was made and the first set of cells were discovered using microscopes in the 17th century. This set off series of studies tilted cell biology, which Robert Hooke pioneered. Years after, scientists started pondering on cellular regeneration and the place of the cell as a fundamental unit of life. In 1839, scientists like Theodor Schwann and Matthias Schleiden formulated the cell theory. However, other scientists also contributed to the theory, like Rudolf Virchow. Till date, cell theory forms the foundation for the fundamentals of biology. It stands as the most widely accepted explanation of the various functions of cells in both plants and animals. The tenets of cell division. There are 3 tenets of cell division, and they are described below. First, all living things are made up of one or more cells. Second, the cell forms the basic unit of structure and organization in organisms. 3. Cells arise from pre-existing cells. The first tenet had been counted based on the fact that non-cellular lives, like viruses, are being disputed as a form of life. Later, the modern version of cell theory came to the fore, and they are highlighted below. First, energy flow occurs within cells. Second, heredity information is passed from one cell to another. Third, all cells have the same basic chemical composition. Advent of microscope. Microscopes enable the discovery of cells. The first form of the microscope was made by the Romans in the 1st century BC. In the 1590s, Zacharias Janssen, a Dutch spectacle maker, started testing various lenses, which form the basis of microscopes as it is known today. As of then, he was able to get 9 times magnifications of the cells. He and his father built a compound microscope in 1595. Robert Hooke was among the first set of scientists to use the compound microscope for scientific discoveries in 1665. Anton van Leeuwenhoek, a Dutch draper, made a simple microscope in 1668 which performed better than every other before it. Electron microscope came to being in the 1920s, which enabled scientists to view objects that are less than one wavelength. Cell cycle. The cell cycle is the progression of the morphological and biochemical events and phases occurring during continuous nuclear replication or cell replication events in a cell. The main canonical processes involved in cell replication include replication and segregation of the cellular genetic materials. Cell division culminates to the production of 2 daughter cells, which are both similar to each other and the parent cell. The cell cycle in prokaryotes is divided into 3 periods, which are the B, C, and D periods. The B period starts from the moment cell division ends to the point at which DNA replication starts. The C period, on the other hand, involves DNA replication. The D period is the period when the parent cell splits into 2 daughter cells. Cell division is classified into 3 phases in eukaryotes, which are interphase, meiotic phase, and cytokinesis. The cell grows during interphase, and it also accumulates nutrients required for the mitotic process. The chromosomes are separated during mitotic phase. The cytoplasm and the chromosomes then separate into 2 daughter cells during cytokinesis. Cell cycle checkpoints are control mechanisms put up by nature to ensure the cell division process progresses normally. Phases of cell division. Cell division is made up of 4 phases, with one phase being distinct from another. These 4 phases are the G1 phase, the S phase, the G2 phase, and the M phase. The M phase is divided into the subphases, which are the mitotic phase and cytokinesis. Proper progression and completion of the previous phase determine the activation of the next phase of cell division, which means the next phase will not occur if the previous phase does not complete. A cell in GO phase is one that has stopped dividing. GO phase. This is the phase in which the cell is out of the cell cycle and has therefore stopped division. G1 phase. This is the phase in which the cell increases in its size and also prepares for G2 phase. In this phase, everything is made ready for the process of DNA synthesis. The G1 checkpoint control mechanisms monitor this process in this phase. S phase. This is the synthesis phase, and DNA replication takes place here. The three phases above make up the resting phase. G2 phase. This is the period of the gap between mitosis and DNA replication. The cell continues to grow in this phase. The purpose of this phase is to prepare everything required in preparation for mitosis. The G2 checkpoint control mechanism controls activities in this phase. M phase. This is the stage of cell division or mitosis. The cell stops growing at this phase and focuses its energy on how to divide the cell into two daughter cells. This phase is controlled by a metaphase checkpoint control mechanism, which makes sure entire cell division takes place. Mitotic phase is shorter than the interphase. It only lasts for a maximum of 2 hours and can be shorter than this. Once one cycle is completed, the daughter cell enters the interphase of another cycle and goes through the entire process again to produce 2 new daughter cells. Each of the stages of cell division is made up of specialized biochemical processes, and these processes prepare the cell for the initiation of cell division. It must be noted, however, that there is no morphological difference between each stage of cell division. Regulation of cell cycle. Regulation of cell division is paramount to the survival of the cell. These regulations detect any genetic damage on time and bring about the repair of the damage. It is also involved in the prevention of uncontrolled cell division, which is the foundation of cancer cell growth. The regulation involves directional and ordered molecular events, which means each of the processes involved occurs sequentially and the regulation cycle cannot be reversed. CDK. Cyclin and cyclin-dependent kinases, CDKs, are the two major classes of regulatory molecules involved. They are the ones that determine the progress of a cell all along the cell cycle. Cyclins are responsible for forming the regulatory subunits. CDKs, on the other hand, form the catalytic subunits of an activated heterodimer. CDKs are inactive when cyclin is absent, and cyclin, on the other hand, do not have any catalytic activity. The CDKs carry out a process called phosphorylation when they bind to cyclin, and this activates the target protein to bring about entry into the next phase of cell cycle. There are different forms of cyclin-CDK combinations, and they are the ones that determine the protein target downstream as the cell cycle process proceeds. Cyclins are produced at different stages of cell division, while CDKs are produced in the cells. These productions occur in response to different molecular signals. MPF. It is the acronym of Maturation Promoting Factor, and it includes both the cyclin and CDK discussed earlier. They trigger progression of the cell cycle into the next phase. P53. It is a protein which functions to impede the progress of cell cycle if any form of damage is noticed in the DNA. The protein can prompt apoptosis, programmed cell death, if the DNA damage is very severe. In damaged cells, the P53 level is increased. It hinders the process of cell division to give the DNA adequate time for recovery. Cancer usually occurs when a P53 mutation occurs. A good example of this situation is in Li-Fraumeni syndrome. P27. This is also protein, and it binds CDK and cyclin, which leads to blockade of the cell cycle from entering into the S phase. High level of P27 has been linked to low risk of breast cancer and vice versa. In cancer cells, there is no control of cell division processes due to the absence or deficiency of regulation molecules. Cancer cells tend to develop faster than normal cells, and these uncontrolled cell growths can lead to metastasis, a situation in which the malignant cells spread from its primary origin to other areas of the body. Photosynthesis. Photosynthesis is the process whereby plants manufacture their foods from carbon dioxide and water to yield glucose and oxygen. The oxygen is then given off as a byproduct. The released oxygen is used by humans for respiration. Photosynthesis requires the ultraviolet ray from sunlight for the process to take place. This means photosynthesis do not occur in the dark. The process involves the conversion of light energy into chemical energy, which is organic molecules like glucose produced in the process. The reactants, carbon dioxide and water, are gotten from the environment. Humans release carbon dioxide as a byproduct of aerobic respiration, and plants take up the carbon dioxide for photosynthetic process. The process of photosynthesis occurs in the chloroplast, which is one of the organelles in a plant cell. The sunlight is captured by the plant via its pigments, especially chlorophyll. The pigments are located in what is called the thylakoid membranes. The space between the thylakoids and inner membrane of the plant cell is called stroma. Purpose of photosynthesis. Photosynthesis is carried out for the purposes highlighted below. First, it counterbalances the concentration of carbon dioxide produced as byproduct of aerobic respiration by animals. Second, it converts energy from the sun, solar energy, to chemical energy, which is used by the plant as energy source for some of its processes. Third, it generates organic molecules that are useful for the growth of the plant. Fourth, it enables the plant to produce food for animals. Fifth, it yields oxygen as a byproduct, which is used for aerobic respiration by animals. Stages of photosynthesis. Photosynthesis occurs in two phases or stages. The two of them are linked by NADPH and ATP. Both stages occur in the chloroplast. The first stage is called the light-dependent reaction, and the second stage is called the light-independent reaction. First, light-dependent reaction. The chlorophyll absorbs energy from the sunlight and then converts this energy into chemical energy. The chemical energy is in the form of NADPH and ATP, which are called electron carrier molecules. The light energy is then harnessed in both photosystems 1 and photosystems 2, which carry both presence in the thylakoid membranes in the chloroplast. Second, light-independent reactions. It is also called the Calvin cycle. This is because the process is cyclic. Carbon dioxide forms carbohydrate molecules with the aid of the chemical energy earlier derived from the light-dependent reaction. This means the light-independent reaction depends on the light-dependent reaction to function. This reaction can occur during both the day and the night. The energy derived from the earlier reaction is used in forming new molecules of carbohydrate. The energy carrier molecules then go back to the light-dependent reaction after it has delivered its energy to the light-independent reaction. This way, it will obtain more energy for another cycle of processes. Photosystems. This is the process involved in the conversion of light energy to chemical energy. The process occurs in a photosystem, which is a multi-protein complex and is present in the thylakoid membrane. There are two photosystems, photosystem 1 and photosystem 2. Both of them are involved in capturing energy from the sunlight, and they do this by energizing electrons. Energy carrier molecules like the ATP and the NADPH carry the excited electrons to power the light-independent reactions. Photosystems are made up of a reaction center and a light-harvesting complex. The pigments in the first process transfer light energy to chlorophyll, molecules located at the reaction center. The light coming in will excite one of the electrons from the two chlorophyll, a pair, and pass it to the primary electron acceptor. The energized electron needs to be replaced afterward. The electron in photosystem 2 is derived from splitting water and releasing oxygen as a byproduct. On the other hand, chloroplast electron transport chain produces the electron required in photosystem 1. The glucose generated from photosynthesis forms the organic compound present in plants for the production of starch, cellulose, and they also participate in cellular respiration. The amount of glucose or starch generated during photosynthesis differs from one plant to another. This amount is estimated in the laboratory using what is called iodine test. The amount of carbon dioxide being released by animals into the atmosphere and which is used up by the plants of photosynthesis is estimated using the phenol red indicator in the laboratory. Different species of plants carry out the process of photosynthesis differently. However, the process always starts with the light absorbed from the sunlight, and it always involves the two stages described earlier. Aside from plants, algae and cyanobacteria are also involved in photosynthesis. Some bacteria that participate in photosynthesis make use of the reverse Krebs cycle instead of the Calvin cycle. The oxygen produced as a byproduct by the photosynthetic cyanobacteria contributes to the oxygenation of the earth directly. Photosynthesis takes place in the chloroplast, as hinted earlier. The chloroplast is made up of certain organelles that contribute to the process. These organelles are highlighted below. First, plastoglobule. Second, plastidial DNA. Third, ribosome. Fourth, starch. Fifth, thylakoid. Sixth, granum. Seventh, thylakoid membrane. Eighth, thylakoid lumen. Ninth, stroma. 10th, inner membrane. 11th, intermembrane space. 12th, outer membrane. Photosynthesis in cyanobacteria. The proteins responsible for trapping light in photosynthetic bacteria are located in the cell membrane. The membranes can, however, be modified into vesicles or thylakoids. The vesicles are called intracytoplasmic membranes. The modified cell membrane increases the surface area of the cell, and this culminates to the bacteria trapping high amount of light. Photosynthesis in algae and plant. Photosynthesis occurs in the chloroplast in plants and algae. There are up to 100 chloroplasts in a typical plant cell, and these chloroplasts are enclosed by membranes which are made up of intermembrane space between the phospholipid outer membrane and phospholipids inner membranes. The plant can absorb light using chlorophyll, and it can also absorb light using other pigments like xanthophylls and carotones. Algae also make use of chlorophyll like plants in trapping sunlight. However, algae also contain series of pigments aside from chlorophyll. Some of these pigments are highlighted below. First, green algae, xanthophylls, carotenes, and phycocyanin. Second, red algae, phycoerythrin. Third, brown algae, phycoxanthin. Factors affecting photosynthesis. The three main factors that affect photosynthesis are highlighted below. First, temperature. Second, carbon dioxide concentration. Third, light wavelength and irradiance. Light wavelength and irradiance. The intensity of radiance of light differs from one location to another. While some areas receive a high amount of sunlight, some other areas do not. Those plants that receive a high amount of sunlight can assimilate carbon dioxide better and faster than those plants that do not. The rate of carbon assimilation tends to increase as irradiance increases. It, however, reaches a plateau when higher irradiance is attained. This increase occurs at constant temperature. The rate of carbon assimilation does not, however, increase at low irradiance, even if the temperature is increased. This is because photochemical reactions are not affected by temperature. However, the rate of carbon assimilation can be affected by changing temperature. Carbon dioxide concentration. The rate of manufacturing sugar will increase as carbon dioxide concentration increases. The increase can be forced to stop by other factors. The enzyme capturing carbon dioxide in the light-independent reaction is called rubisco, and it has a strong affinity for oxygen and carbon dioxide. It will bind to oxygen when the rate of carbon dioxide is low and will bind to carbon dioxide when the latter's concentration is high. This is a process called photorespiration. Cellular respiration. This is a combination of metabolic processes and reactions that occur in the cells, and it is involved in the conversion of biochemical energy to ATP. The biochemical energy is obtained from nutrients. The reactions involved are all catabolic reactions. This means large molecules are broken down into smaller ones in the reaction. The process leads to energy release. The reactions are among the major routes via which cells released chemical energy which act as fuel for cellular activities. It is an exothermic redox reaction since it produces heat as byproduct. The reaction involves several biochemical steps. These series of reactions can generate heat very slowly. Some of the nutrients used for the process are fatty acid, amino acids, and sugar. Oxygen stands as the most common oxidizing agent in the reaction. The energy is stored as ATP, and ATP can be used by the cell to power various processes like transportation of molecules across the cell membrane, locomotion, and biosynthesis. Aerobic respiration. It is a respiratory process requiring oxygen which is used in producing ATP. The reactants are proteins, fats, and carbohydrate. The end products are water and carbon dioxide. Oxidative phosphorylation is used in making most of the ATP that is produced during the aerobic cellular respiration process. By normal standard, aerobic oxidation can produce up to 38 ATP, 34 molecules from electron transport system, 2 molecules from Krebs cycle, and 2 molecules from glycolysis. However, certain conditions prevent the attainment of this huge amount of ATP. One of such conditions is ADP and pyruvate into the mitochondria, and this can cost between 29 and 30 molecules of ATP per glucose moved. Another limiting factor is the existence of leaky membranes. Aerobic respiration is more efficient than anaerobic respiration. The former is 15 times more efficient. Anaerobic respiration can only yield 2 ATP, while aerobic respiration can yield up to 38 ATP. Some anaerobic agents, like methanogens, can yield more than 2 ATP via anorganic molecules. The points highlighted in this write-up form are just a part of the many fundamentals of biology, both in plants and in animals. Undoubtedly, biology is the widest field of study in all human endeavours since it encompasses both plants and animal studies. There is also one another aspect of biology called microbiology, which studies unicellular organisms, some of which have been mentioned in this write-up. Humans are not resting on their oars. Research is ongoing and many more aspects are being introduced into biological sciences. One of such new introductions is genetics. An even newer aspect is molecular biology. There is a meeting point between molecular biology and genetics, with the latter forming the basis on which the former is built.